So we're going to move from DNA to protein, and we're going to have a couple of talks about biomarkers, which are near and dear to many people's hearts, at least in this audience. Our next speaker is Bill Romm, who is a professor of environmental medicine and medicine at NYU. He has been working for quite some time on lung cancer and trying to develop tools that would allow earlier detection of lung cancer, which could be of great clinical significance.
Thank you very much, and thank you, Larry, for the invitation. I think one of the more important things today is this gene-environment interaction, and I think Richard, Dr. Spritz, really started us off well with the gene part. I'm an environmentalist. I'm going to talk about environmental medicine. I will get to proteins and biomarkers, but I want to encourage any students who are sitting in the back row that I was one of you in 1963 here at the University of Colorado. I did major in skiing. I did do 100 days on the slopes as a sophomore. I graduated on time in '67 and thought I would be targeted and reach all those forks in the road, but I really didn't know what to do, so I took the medical college admissions test, and just in case, I took the law school admissions test, and just in case, I took the GRE so I could get a PhD in political science too. And I went to medical school, and I've been a student ever since those days, and it's a wonderful life. So I hope there are some students here and you can charge ahead. So I'm going to talk about one of the major pulmonary diseases. I'm a pulmonologist, an internist, and I went into pulmonary medicine because I love the environment. It's close to air pollution, asthma, COPD, particularly asbestos in occupational medicine, where asbestos causes asbestosis or fibrosis and pleural plaques, but it also causes lung cancer and mesothelioma. I did my fellowship under Dr. Selikoff at Mount Sinai in New York, who was the father of environmental medicine. So I really got a treat in learning and doing science about environmental factors that cause disease. So lung cancer is our topic for today, and I'm going to use this as a disease, as an example for environmental exposures, and an example where biomarkers can make a difference, and where Larry Gold can make a difference in personalized medicine. So this is a major disease. There's 159,000 deaths in our country, 219,000 cases in 2009. It exceeds the deaths from breast, prostate, colon, and kidney combined. It's the leading cause of death for women. And the non-smokers are a major group in this, probably 20 to 30%. So there are 1.2 million lung cancer deaths globally, so it's a major disease not only in the developed countries, it's a major disease in developing countries like China, where all the men smoke practically. It costs a lot of money. CDC estimates public health costs to be $5 billion a year. Treatment costs are double of that. The problem with this disease, like many diseases we as internists see, is that the disease is advanced and spread by the time they come to you complaining of a cough or shortness of breath or chest pain. So this is a disease that has metastasized or spread to regional nodes in the mediastinum or distal sites like liver, bone, brain, and 3/4 due to late diagnosis. And there's a lot of histologic heterogeneity with this disease. It's not just one disease, it's a whole variety of diseases, so it exemplifies the genetic complexity of disease. And then half of all cases are diagnosed in ex-smokers, 15% in never smokers. So I don't have any conflicts of interest, but I do get funding from the NIH. I'm funded by the NCI for this early detection research network. I'm also funded by Heart, Lung, and Blood for research on AIDS and TB and pneumonia. And I'm also funded by the National Institute of Environmental Health Sciences because I train doctors to be environmentalists like myself, so I have a training grant. So the NCI Early Detection Research Network is an extramural program to develop biomarkers for the early detection of cancer. There are about 35 grantees in this program. There are 10 centers that are epidemiological centers for collecting samples, and we have one of these at New York University that is my grant. We call it the NYU Lung Cancer Biomarker Center. There are 24 biomarker development laboratories. We have several of these at NYU and osteopontin for mesothelioma and glycomics. And then there are some reference laboratories in a center for statistics. This is a U grant program. Those of you who are going to be funded by NIH, you want to have R01s because those are your tickets to success. They're individual research grants. But the NIH now has a series of grants called U-Series. And U-Series are double the work. They have you go to 3 meetings a year. They have you present 3 times, and to try to generate data to present 3 times a year is a real challenge. And they force you to speak to other people so you become collaborative and not isolated. So it's a interaction between NCI staff and the grantees. So we have steering and executive committees and collaborative groups for breast, prostate, GI, and lung and upper aerodigestive cancers, of which I chair the latter group. So I said this was going to be the environment part of the gene-environment interaction, and for lung cancer we have a very major environmental or personal even, personal medicine exposure of cigarette consumption. And cigarette consumption has been a boom industry, as you can see in this slide, during the 20s, 30s, 40s, and especially during World War II. As you can see that we've had an increase in cigarette consumption, but we hit a peak with the first Surgeon General's report in 1964 showing that cigarette smoking caused lung disease and heart disease, and people began to wake up that this might be bad for your health. Following that, there were smokeouts, ad bans, fairness doctrine, but the major influence on reducing cigarette smoking was the federal cigarette tax, which doubling of that and increasing cigarette taxes in the states reduced the cigarette smoking incidence substantially. Now, on the bottom of this slide, the U.S. cigarette smoking rate increased from 19.8% in 2007 to 20.6% in 2008. We haven't won against this exposure yet. And I also want to point out that cigarette smoke isn't the only exposure for lung cancer. We have air pollution. Particulate matter of 2.5 is associated with cigarette smoker with lung cancer, and also asbestos, radon daughters, and so on and so forth. So this is a considerable environment-gene interaction. The next thing about lung cancer is that this is a chronic disease that evolves over years, and there's a concept called field cancerization. So when you smoke, you have the tobacco going throughout your respiratory tract, and you have changes that are genetic occurring in the bronchoepithelial cells in particular. And these may be a patch, like on this slide, or a field, and then a carcinoma. And then as you go through this change, you get a hyperproliferation of the cells, and you accumulate chromosomal and genetic defects. This occurs not only in squamous cell carcinoma in the bronchoepithelium, but we have the same pathway in adenocarcinoma, which is in the peripheral airway, right at the bronchoalveolar junction cells. So our gene friends have been sequencing lung cancers. This is a series of 188 lung adenocarcinomas that were sequenced with one of the sequencing projects of the NIH, published in Nature in 2008. And you can see that there are many mutated genes in lung adenocarcinoma. There were about 25 major ones, but it seems like there were some that were found in practically almost all of the tumors, like 60 to 70%. These were in well-known cancer genes. One was called the P53 gene, that's the one on the far left, then KRAS, which is a signaling gene, then a serine threonine kinase gene, and then a receptor for a growth factor, EGFR. So this is more scientific jargon, and rather than leave you with that, I'll talk a little bit about these genes and these pathways and try to define them so you can understand them. So P53 is, first of all, a tumor suppressor gene, and it's the most commonly mutated gene in cancer. It is a transcription factor that activates genes involved in DNA repair. So what's a transcription factor? So a transcription factor is a protein that binds to the promoter region of genes and activates them and turns them on. So that's fairly simple. But now with all this omics coming out, we now have transcriptomics. And transcriptomics is a whole lot of transcription factors, but each transcription factor can bind a whole lot of genes and turn on a lot of genes. So understanding this has now become another huge omics science. And there is technology from ChIP-seq and RNA-seq and all these various seqs to identify the sites and the areas of genes that are bound to transcription factors. So all of a sudden we realize that this is pretty important, and if it's involved in cancer, it's really important. So one of the genes that p53 binds to is p21. And this one is called a checkpoint control at G1S. So on the previous slide, I showed you that cancer cells begin to proliferate more rapidly. So they have an independent growth control. So they're involved in the cell cycle. So cell cycle is really important for cancer because these are cells growing much more rapidly than normal cells. P21 is a checkpoint control to stop the cell cycle. And this is something that when P53 is activated, it activates this gene to slow things down so we can repair the DNA damage leading to cancer. And the next gene is GAD45, is one of these DNA repair genes. So P53 has an important function in fixing things like tobacco smoke is causing DNA damage in the lung. So if this gene is mutated, that means it doesn't work in preventing cancer. So that's critical. And if P53 can't slow things down so that other genes can't get in there and fix the lesions that leads to cancer, P53 can turn on a pathway called apoptosis. And this occurs in cells that express oncogenes and inappropriately bypass the G1S checkpoint. Apoptosis is another jargon that's really important because that means you can kill the cancer cell and get rid of it. So if P53 can do that, that's really important. But if P53 is mutated and can't do that, you end up with lung cancer. So the other thing, before the omics era came along, just before that, in maybe the 90s, if you made a mouse, a transgenic mouse, you got a science paper or whatever. And so everybody in the world of science was making a mouse. And that was pre-omics. So we made a mouse. So here's our mouse. And this is a mouse that has what's called a CCSP promoter. And that stands for on the top of the slide, that stands for Clara Cell Secretary Protein. So this is a promoter that will make your gene, that whatever you're putting in, be expressed only in Clara cells, which are only the airway cells in a mouse. So we can make this transgenic mouse express our abnormal gene only in the lung. So we want that. And then in that mouse, we put a dominant negative P53 gene. That's the DNP53. And what we did there is we took out the DNA binding portion of the P53 gene so it wouldn't work. So it's a dominant negative. But it maintained the dimerization tail of the gene so that this dominant negative would bind all the endogenous P53 in the lung and prevent it from working. So we'd have a P53 ablated lung mouse. We made our mice, and we had two mice, 2501 and 3503, and you can look down here on the bottom under lung. On the lower one, the band shows a strong expression in the 3503, so we expanded that mouse, and we then looked at some of the lungs, and we could see that there was CCSP staining to the antibody in brown in the upper left and P53 antibody in the upper right, only in the epithelial cells of the lung. And you can see tumors forming, and we were able to form these tumors in these mice. So here is our results. We had 20 wild-type mice, 31 dominant negative P53. We had 20% tumors in the wild-type, 45% in the dominant negative P53. We had an odds ratio of 3. So we were very happy, except for that. And everyone who's a scientist here knows that you want that to be less than 0.05 to get your paper published. So we decided that we had to do something to get this below 0.05. So we exposed these mice to intratracheal benzoate at 4 milligrams, and then waited until they were 9 months old instead of 18 months. And the benzoate treated DNP53 mice, 73% got tumors compared to none in the solvent-treated wild type, with an odds ratio of 25 and highly significant. So benzoate, more jargon. This is the stuff that's the carcinogen in tobacco. This is tar. Okay, so everybody's with me? So these mice got cancer very rapidly, and we showed that a tobacco carcinogen would accelerate the genetic defects, a gene-environment interaction. Everybody's with me? Okay, we looked at PCNA. This is a proliferative marker, and you can see in the tumor that all these brown staining cells show that there's a lot of proliferation. This is high power. But look here, this is sort of normal lung right down here, and here's some cells that are beginning to look a little abnormal. So here's some increased proliferation. So this is an early lesion. So here we have what we think is going on in humans, where you smoke and you accumulate these chromosomal mutations, genetic mutations, and then all of a sudden you begin to get a clump of cells that have enough of these mutations they can form a clone. So here is the early development of lung cancer, and this is the disease that you want the surgeon to take out. This is the one you want Larry Gold to develop a protein biomarker for, so you can diagnose this disease. So back to P53. So P53, as I said, is a key gene for lung cancer. And this is P53. This is DNA that it binds to. But you'll notice that there are, of its 330 amino acids, there are some labeled in yellow that bind to the DNA. So amino acids at 248, 249, 245, 175, 273, 282 are all the binding regions to DNA. And you remember in the mouse, we cut out the central part of the gene. We cut out from 15 to 300. So we cut out all of these binding areas. Now, what if you smoke and you form DNA adducts at these binding regions, and your smoking would prevent P53 from working, and maybe these would be the sites that would become mutated? So a DNA adduct, more jargon. So benzoate binds to these transcription factors, to proteins, and forms adducts that prevents DNA replication. So this is looking at the P53 mutations in lung cancer. And you'll notice a difference between non-smokers and smokers. You know, the mutations are throughout the P53 gene in non-smokers, but look at smokers. They occur at 157, 248, 273, and radon-associated at 249. These are called the hotspots of P53. And I have a colleague at NYU by the name of Eric Tang, who's an expert basic scientist who does gels with P32 to look for adducts. And he's been looking at benzoate, but he also got interested in acrolein. The problem with benzoate is one of dose. There's about a microgram or less in a cigarette. But acrolein is much more abundant in tobacco smoke, 1,000-fold more abundant, 40 to 50, these should be micrograms rather than milligrams, micrograms per cigarette. Acrolein is an irritating aldehyde and an air pollutant from fossil fuel combustion. So this is not only in tobacco smoke, but it's also in vapors from hot oil and cooking, exposing women in particular, particularly the Asian women. So you can get exposed to acrolein, which is an irritating aldehyde, from these hot woks and whatever that women have to cook around all day. So the question is whether acrolein can form these DNA adducts. This is a gel that students used to have to do, but now you just do the whole microarrays instead of having to do gels. But this is a P32 gel looking for these DNA adducts in exons 5, 7, and 8 of P53. And this is exposure to acrolein up here and benzoate down here. And these are the 3 exons. And on the right-hand side are fibroblasts and bronchoepithelial cells. So we'll just look at the cells that were exposed in vitro to either acrolein up here or benzoate down here. Just focus your eyes on these numbers in the 150s, the 248, 249, 273, 281, and the same here with benzoate. These are adducts. These are DNA adducts, and the point is they're occurring at all of these hotspots. And this is a comparison of the adducts and the mutations on exon 5, adducts and the mutations, adducts and mutations, and this was published in PNAS. Now this is a complicated slide. I was going to take this out of the talk, but I left it in for one reason, and that's because this first came out in Science in 1999 with just the benzoate, and it showed that tobacco smoke contained a carcinogen that definitively and genetically caused damage to cause lung cancer. And this came out right when the attorney generals of 44 of our 50 states were suing the tobacco industry. And the tobacco industry then capitulated, agreed that they needed to do something, and the master settlement agreement was formulated, committing about $240 billion over 25 years from the tobacco industry to these states for supposedly for health care, but as we all know, this money has been going to fill potholes left and right. But anyway, so this is important. Science has important policy considerations. Now, I mentioned that way back when that the second gene was RAS, and that there were other genes involved in signaling called EGFR, epidermal growth factor receptor, and that all of these signaling genes are involved in causing events that lead to cancer. So growth factors would come from the environment as ligands, bind to these receptors, stimulate RAS, PI3K, and these signaling pathways through the MAP kinases, and then you would have proliferation of cells, the cell cycle would be stimulated, invasion would occur, metastasis would occur, resistance to apoptosis, and angiogenesis as you develop a clone of cells, and you'd have angiogenic factors stimulated. So this whole slide explains many of the events that occur across individuals in cancer pathways, not only for lung, but other cancers as well. So we'll put a few postdocs on this cell cycle, and I'm going to present a few postdoc projects. And here's one where we took a receptor, not the EGF receptor, but the IGF-1 receptor, and we made some mutations in it, and then put this receptor into A549, which are lung adenocarcinoma cells. And with this mutant receptor, we were able to take the cancer and turn it into a glandular differentiation. And the mucin that wasn't in the cancer would be seen here, and this would be an HA tag. And we could eliminate all of the apoptosis that we would see in the rapidly dividing cancer cells. So by just changing some of the pathway, we could eliminate or change the cancer phenotype. So that's what we did with the IGF receptor, similar to the insulin receptor, but there's this whole signaling pathway, and we interrogated RAS and the MAP kinases, since this is important in the proliferation pathway. And I wanted to show you what some of our fellows in pulmonary disease at Bellevue Hospital do, since they have a research year. So this is one of our fellows who bronchoscope, and I want to tell you that translational medicine is very different than working in molecular biology, because you have to deal with something called IRBs, institutional review boards. So you have to get a project through them. And that's only the beginning of your misery. Then you have to find patients. And once you find a patient, then you have to get in consensus, no problem. But then you have to do something to get the sample. And in this case, we bronchoscoped them and lavaged them to get the cells, and then also did a transbronchial biopsy. This is really indicated clinically, but sometimes we do these for research purposes, and that's an added complexity of work that you have to do before you get your specimen to do your molecular biology assay. So we got the cells, and then we looked for a PCR assay looking for mutant RAS. And since this is a codon 12 mutation, a single mutation in the gene, it was fairly easy to find it with PCR. But half of our adenocarcinomas had this mutation, 14 out of 25. And you can see we didn't see it in squamous cell, and we didn't find it in any of the other diagnoses. So it was fairly specific for adenocarcinoma. We then went to Eric Tang and asked him to see if adducts formed on this from benzoate-pyrene. As you can see here, he took cells that were bronchoepithelial cells in vitro and added benzoate-pyrene and formed these adducts at codon 12 that I mentioned to you. But I wanted to explain to you this problem, adducts on codon 14, where we do not see any mutations in humans. So why did he get these adducts? So he then tried to figure this out in his laboratory, and he took the cells and cultured them for 24 hours and looked at the loss of these adducts over 24 hours and found that in codon 14, in the KRAS gene here in the yellow, as he followed these cells out for 24 hours, the percent remaining adducts were basically zero. They all disappeared. And the same thing happened for NRAS and HRAS. But look at this data point. This is codon 12. 50% of the adducts were remaining at 24 hours, suggesting that there's something lacking in getting rid of these adducts. So there's a repair problem as well. So that may explain why we see this at the codon 12. And I also mentioned that below RAS was the MAP kinase enzyme. And we had a junior faculty person who had one of these K grants from NIH where you get 5 years of salary. So she was able to do phosphorylated P38 assays on a whole series of lung tumors. And here was a lung that was taken from the patient that was normal lung, uninvolved by tumor. And then this is the tumor, and you can see that this enzyme was increased. This is the control. And the tumor was increased in all 6 of these patients compared to the lung. So we then went through 19 lung cancers, and this is the increase in the phosphorylated P38 in all of these tumors, and 18 of 19 were all in the right direction. But when you do science in humans, you know, there's always an outlier. We couldn't figure out why this one didn't work. So I asked her, you know, what's going on with this patient? And she went and looked and found out that that was a specific metastasis from the colon to the lung. So that really was a whole different ball game. So we think that's why that enzyme wasn't increased and that this is specific for lung cancer. So we went to our pathology lab and we had immunohistochemistry showing that from normal to the upper right where you have dysplastic to the lower right where you have cancer, you have increased immunohistochemistry staining for that enzyme. And we then start putting together this idea of what's going on, similar to the previous graph that I showed you, that you have ligands binding to receptors, TGF-beta, EGF, acrolein making these adducts and mutations. But we also found that TGF-beta receptor is reduced, and that blockage of inflammation would increase NF-kappa B to be activated, that KRAS would signal to this, that all of these P53 mutations would prevent apoptosis, would prevent the block on proliferation, and that NF-kappa B affected all of these events from inflammatory cytokines to angiogenesis to cell adhesion to anti-apoptosis to proliferation, so that there's an interaction between inflammation and field carcinogenesis for lung cancer and many other cancers as well. So we've taken the inhibitor to NF-kappa B, the inhibitor kappa B beta, and we added it to A549 lung cells again. And then here we did different levels of serum with the cells and then added IGF-1 in these wells. And you can see that the vector alone, that these anchorage-independent assay would form a lot of cells. When we added the inhibitor, you can see how this is much reduced and blocked in comparison to the vector with the no inhibitor to the NF-kappa B. We took these cells and then we injected them in a model of lung cancer metastasis where we had A549 cells injected in the tail vein, and all of these lungs had metastases in them. When we put the inhibitor in these cells and injected them, you can see there's far reduced metastasis. So that kind of shows you some of the influence of inflammation on carcinogenesis. So one idea we have is we've been treating multi-drug resistant TB with aerosolized interferon. We then did a randomized control trial in Cape Town, and then we had the idea, why not try this for these small nodules for lung cancer and see if we could prevent it. And we added interferon to A549 cells in vitro, and you can see it inhibits this transcription factor at 24 and 48 hours. So that is a trial we're planning to write up and develop over the next year or two. So in lung cancer, you've heard that there's now CT scan screening. So why do any of this biology? You can just go in and get your whole body CT scan, look for coronary calcification, et cetera, and get a whole body scan and pay your $1,000 and see if you're okay. So we have been enrolling heavy smokers in a CT scan project at NYU, but we also collect blood and get sputum for biomarkers. And this is what we see. So this is a patient who was a heavy smoker, came in, and this is a cut through the lung. And all of a sudden, the radiologist puts an arrow on that, and you begin to get worried. And you look back, and did you miss something right there? And was something growing there? And then you tell the patient to keep coming in, and this thing keeps growing, and the patient says, I refuse to go to the surgeon. And finally, we got this patient to go to the surgeon. Yeah.
You go back.
Oh, okay.
So will you, those of us who don't know how to look at these, I don't, I would be just as worried at the picture on the left as the one on the right, that one over from where the arrow is.
Okay.
So what is it about those two scans that made the guy put an arrow where he put it?
All right. So Larry would prefer to be a radiologist. So this is a blood vessel looking straight at you. So there's blood in there, so that's more dense. This is straight at you or away from you. So that's why you see these. And the reason you know those are blood vessels is you see the blood vessel coming off it. And you see a blood vessel coming off this one and off that one and off that. Okay, right here you have a something that's a little bit bigger than these blood vessels, and you don't see any blood vessel coming off it, and it's more peripheral. So CT scans pick up these peripheral lesions. And you go and look over here, and I mentioned that there's maybe something here, and I'm not sure about that, and there's this, and maybe that's a blood vessel. So this is at a point you really are not sure whether that's anything. And you go way back here, and oops, there's nothing there, and there's something there, and there's something there. So it might have been growing. And then here, it's a little bit too big to be acceptable. And now you can see it's really getting big, and the surgeon's got to get that out of there. All right, everybody a radiologist. There's a worry factor here. All right, so that's why we need a biomarker test to help us with our worry factor. And so this is for nodules on the CT that are 1 cm or less, or something that we call GGOs. And so I better explain jargon. GGO is ground glass opacity. And GGOs are when things look fuzzy. And we see a lot of GGOs on these CT scans. And if they're 4 millimeters or less, we forget about them. But when they're 4 to 8, we get worried. When they're bigger than 8, we get really worried. And we call them back for frequent visits, and we try to figure out if this is a cancer or not. So I've got 1,200 of these high-risk smokers, and we're worried about these nodules because we want to make early diagnosis, we want to avoid missing a cure, we want to avoid futile thoracotomies, and we want to avoid anxiety, especially of the physician like me. This is our 1143 lung cancer biomarker center screening individuals. Blue are the screening. Red is those that come in and they're part of our surgical group because they already have nodules. The surgical ones are a little bit older. Most of the biomarker screening individuals are in their 50s and 60s. And we enroll people with more than 20 pack years of cigarette smoking. So most of the blue are heavy smokers. They have a mean of 43 pack years. That's 1 pack per year. So most people smoke 1 pack a day. If you do that for a year, that's a pack a year. If you do it for 10 years, that's 10 pack years. So 43 pack years is a lot of cigarette smoking. And it's very hard to smoke less than a pack a day because you got to keep your nicotine up. Nicotine lasts only about 19 minutes as a half-life. So every half hour, you got to light up. Got to get out of this room and smoke in the outside. So half of our patients have nodules, solid nodules or GGOs. That's very worrisome because these are nodules that are larger than 4 millimeters. So CT scan screening has been popularized as a panacea to prevent lung cancer. So there's early lung cancer action project at Cornell that we were part of. It published in Lancet 1999 that they found 27 lung cancers with CT scanning and only 7 were visualized on regular chest x-rays. That was published in Lancet. They then did a national study that was published in the New England Journal finding that they found early lung cancer, but then this was countered by a report in JAMA from Memorial Sloan Kettering. That was a combination of Milan, Italy, Moffitt in Florida, and Memorial, where they screened 3,200 smokers. They found 144 lung cancers with 44 expected. They had a 10-fold increase in resected, but there was no evidence of a decline in the number of diagnoses of advanced lung cancers or deaths from lung cancer. So mortality was not changed. So NIH is spending $200 million to try to answer this question with a national lung cancer screening trial of 50,000 smokers and ex-smokers. They'll be screened with X-ray versus CT scan, and this is should be done in a year. So these are the cancers we found in our screening trial at NYU. You can see we pick up little itty-bitty things, and that haze is the ground glass, the black is the solid nodule. We've now looked at mortality, and this is collaboration that some of our results may lower mortality. If mortality were not lowered, it would be a straight line on this curve so that it's moved to the right. It shows a lowering of mortality. So we're looking for biomarkers, and in the last 10 minutes I'll tell you about biomarkers. So we've looked at autoantibodies in the serum. This is a panel of 10 autoantibodies. The red is lung cancer. The green on the left are smokers. So we've developed an ROC curve of 0.9 of cancer versus no cancer using autoantibodies to the 4 cyclins, survivin, and c-MYC. This is going to be published in BMC Cancer. We have joined with Celera. This is the importance of biotech industry. Celera has done mass spectrometry of proteomics, looking at tissue specimens, cell lines, and conditioned medium for 500 markers that they did a small 12 normal, 12 tumor verification study where they found increased markers in tumors versus controls. They reduced that to 27 markers. Then they got to a 9-marker panel with 104 cancers and 103 controls. And now they are getting samples from myself and Dr. Pass at NYU, where we are doing a secondary validation where we have a 9-marker panel with an area under the curve of 0.977 with 90% sensitivity and 96% specificity. And I need to tell you what those things are because sensitivity above 90% and specificity above 90% is good. And an ROC curve that follows this line is identity. So now you want to go to the left of that curve and you want to get, this is perfection. None of us achieve perfection, but a blue line like that is really good. So this is a 9-marker panel. This is a 6-marker panel. So these are proteins in the serum. This is a training set. The next you do a validation set. These are the markers. Since they're biotech, they have numbers on these, so I don't know what they are. But some of them are well known. So each one of these has a certain statistic, and what they found and what we've all observed is that a panel of a number of these markers has much greater sensitivity and specificity than each individual marker. So SomaLogic has now called SomaMers. with these aptamers where they have scientifically developed these nucleic acid aptamers that bind with tight affinity and longevity with specific targeted proteins. And they're up to 865 of these aptamers. So at NYU, we provided control smoker sera, lung cancer cases sera, and then along with our samples, Pittsburgh and several other sites provided samples so that SomaLogic has had 1,326 cancer cases and controls to run on. So this is their training set, verification set, showing that there's demographic factors that are important. But the exciting part is that there is an ROC curve showing the performance of 12 biomarker lung cancer classifier by the various study sites that looks really good. And this is identity, as I said before. All of this is good when it goes to the left. The black dotted line is all of the various sites. The only important thing here for you to look at is NYU is the light blue. So we were better than all our other sites, so SomaLogic should just come to us for samples. So these are the 12 protein biomarkers. They represent a whole series of proteins that are involved in inflammation that I said before and cell cycle. But the Pittsburgh colleagues did this ingenuity pathway analysis based on looking backwards at what genes might be involved in these proteins. And you'll look at these crazy figures, but IL-13 is an inflammatory marker. Matrix metalloproteinase is an inflammatory marker. This is another inflammatory marker found in the lung. IL-4, TNF, these are inflammatory markers. Here's our friend P53 that I've been talking about. Here's the P38 MAP kinase. So everything I've told you about has been rediscovered by Larry Gold. Another assay you can do on the omics is a genomics assay of the blood. So this is a combination with FV-metrics microarrays looking at blood cells. A 29 gene classifier was used to look at non-small cell lung cancer samples, and these are control samples. So every line is a case, and all these above the line are cases of lung cancer. These are the misclassified lung cancers. This is the controls. Every line below the identity is a smoker control. These are the smoker controls that were misclassified as cancer. This is the ROC curve. So you can do genomics of the blood to try to diagnose cancer. These are all the various genes that came up. This is in Cancer Research from December. And lastly, I want to look at nanoarchitecture rather than just protein assays. So you can go look at this field that's cancerized, and you can actually get cells out of the mouth. And these are buccal cells. And this is looking at partial wave spectroscopic microscopy of cell nanoarchitecture. These are COPD with just regular light up here. This is looking at nanoarchitecture of 3 patients with COPD. And these are 3 patients with COPD and lung cancer. And you can see that the disorder strength changes, even in the buccal mucosal cells. And that's the ROC curves. These are all the people that did all the work. And that's what I do when I'm not here. This is North Cascades National Park up in Washington. And Richard said he was going to a personalized medicine thing tomorrow. I co-chair a climate change and respiratory health meeting. And I was there climbing El Dorado Needle because I wanted to see what the effect of climate change was on glaciers. And our guide said that there were all these bergschrunds that had developed from last year to this year, and the glaciers were melting, but we were able to climb up this needle, and this is jumping off the top with a rappel, and that's what physicians, scientists do in their spare time. Thank you.
We have time for a couple of questions.
Leslie, thanks. The button was necessary. The ROC curves look wonderful. Have you tried to look at the different technologies together? Can you improve on that actually quite good ROC curve even more with, say, putting the RNA technology with the protein technology with the, say, the genotyping?
Yes, so that's a very good question. We have a whole series of patients coming in both for lung cancer and for high-risk smokers. So we collect these samples and we share them with other investigators. Usually, so there's quite a bit of overlap that we send to different groups that we could look at that. We haven't yet because we're just getting the data back from each one of these separate studies, and we haven't had a chance to mix and match. So what we've done is that this grant ends in June, and the new grant begins in July. None of us know if we're going to be survivors or not on July 1. But what we did put in the grant going forward is that we're going to be prospectively collecting these samples, and we want to compare the technologies and do exactly what you said. So I wish you were a reviewer.
Well, God knows I might be.
So I went to here, to your left. I think one of the key observations that you report is that the disease genes are awful biomarkers in this particular case. So one of the conclusions from the previous talk was that if you identify the disease gene, you can do molecular diagnostics. This is clearly not the case for lung cancer. We have p53 and RAS, potent tumor suppressors and oncogenes. They are there in the normal tissue and in the diseased tissue. Yes, they have a point mutation, but your panel of biomarkers did not include p53 or RAS. In fact, there was not a single p53 target gene in that panel biomarker. If you compare the disease genes that you show in your first slide with the biomarkers in one of your last slides, there's basically no overlap. So is this a rare exception where the disease genes are not good biomarkers? Finding biomarkers is going to tell us anything about the etiology of the disease. How much can we learn from disease genes and biomarkers back and forth?
Yes, the question points to all these mutations and changes in these upstream genes like P53 and KRAS. And I think what this investigation points out is the importance of pathways. And that when you have pathways, these genes affect many other genes. And when we see an autoantibody profile of all these cyclins coming up, we know that the cyclins are being overexpressed and that autoantibodies are being made against them. And the cyclins would be tamped down if P53 weren't mutated. So what we're seeing in the serum is end effects of some of these defects that environment-gene interaction promotes in the lung.
Could I ask you one question?
Yes.
At a different level, tied with my lack of knowledge of radiology is my lack of knowledge of the lung, actually. It just seems as though someone ought to be trying to measure stuff in exhaled breath as an early detection for non-small cell lung cancer. Is there activity doing that? Does that make any sense at all?
So I threw out all those slides because I've already had 1 or 2 minutes. So yes, we've done that. And we collect exhaled breath on charcoal tubes, and then we ship them to a mass spec laboratory in New Jersey. That's a small business, NIH-funded research organization called Mensana. And they're very good at analyzing this, and then they do all kinds of bioinformatics and bootstraps and what have you to identify those compounds that may be associated with lung cancer. And surprisingly, they can separate out lung cancer from smoking, which may be a huge confounding factor. They come up with panels of about 20 long carbon chain alkanes that seem to be related to reactive oxidant species and oxidant stress. And the challenging thing of their test is that from site to site and from experiment to experiment, the 20 items are a little bit different. I'm a little worried about that, but they are now moving toward a large validation trial to go to the FDA to get approval to use this as a test.
Thank
Thank
you.
you.